This video will cover the following topics. The DNA replication process involves enzymes. Let's get into the DNA replication process. First and foremost, DNA is double helixed, meaning that there are two strands in each DNA segment. In order to replicate DNA, we have to separate each strand, and each strand will serve as a template for a new piece of DNA, meaning that the new DNA will have one old strand and one new strand. This means that DNA replication is semi-conservative. Different DNA strands also have different directions. They run anti-parallel to each other, which means that they are parallel, just one is basically upside down. We call the ends of the DNA strands 3' and 5', named after how we refer to carbon molecules in the deoxyribose space in DNA. This is important for later. First, we had to uncoil the DNA because right now it's all twisty. So, how do we keep it straight for other enzymes? This is where the enzyme topoisomerase comes in, which prevents supercoiling, or basically just being in that twisted form of DNA that we all know of. Next, we have helicase, which finds and attaches to a specific sequence of bases, named the origin of replication. This is basically where replication will start. It splits the DNA into two separate strands into a replication fork. There are also single-stranded binding proteins, which binds the single strand of this template DNA to prevent it from resealing itself after it is separated by a helicase. Next, we have primase. This, basically like the name, primes the DNA for making new strands by putting RNA primers, short RNA sequences that are basically there for DNA polymerase to latch onto. Most importantly, we have DNA polymerase. This is why primases are needed. DNA polymerases need the RNA primers to know where to start replicating. This enzyme synthesizes new bases by reading the template strand of DNA and attaching the respective base. DNA polymerases synthesizes in the 5' to 3' direction. If DNA is semi-conservative, then there are two different strands being synthesized, but they aren't replicated in the same way. One of them is the leading strand. This strand is synthesized continuously, and its DNA polymerase follows the direction that the helicase is going in. The other is the lagging strand. Because DNA is anti-parallel and has to be synthesized in the 5' to 3' direction, this means that it will have to go in the opposite direction of the leading strand. The lagging strand is synthesized in fragments because it can't be built continuously. Once it attaches onto a primer, the DNA polymerase will go until it hits the last piece of DNA, and then from there, a different primer will prime another spot, and a different polymerase will go behind the last fragment and continue going until it can't. These fragments are Okazaki fragments. That leads us to our next enzyme, ligase. Ligase actually binds the gaps between Okazaki fragments together into one continuous DNA strand. So, what have we learned today? DNA replication is semi-conservative. Topoisomerase uncoils DNA. Helicase splits DNA into strands. Primers put down starting points for DNA polymerase. DNA polymerase synthesizes new DNA. The leading strand is synthesized continuously. The lagging strand is not synthesized continuously and instead in Okazaki fragments. Ligase connects Okazaki fragments together. Thank you for watching. If you have any feedback, please leave it in the comments below. And thank you everyone for watching.